Welcome to solverfy.
So what is the role of plasmids in molecular cloning? This is the overall process sketched out here of how genetic modification or molecular cloning occurs or is done. And if you notice, plasmids are these circular bits of DNA. Okay, so they're just a loop of DNA that are found in bacteria. These loops of DNA sometimes have genes that encode for some sort of protein in the bacterium. And notice that they are separate from the actual genome. So although the genome is also a circular piece of DNA, it is actually so genome is this guy over here. The plasmid is a piece of DNA that is separate from the genome, and sometimes the plasmid can be put out into the environment and picked up by other bacteria, which you create transgenic bacteria that way. Okay, so there's a lot of lateral swapping of plasmids between bacteria. So we've learned to manipulate these plasmids for the purposes of genetic modification. So plasmids have certain sequences in them that these enzymes called restriction enzymes can recognize. So restriction enzymes recognize certain section of the plasmid and cut the DNA right there. Now we can choose the restriction enzyme of interest, cut the plasmid, and then use ligases to insert a different piece of DNA into that same plasmid, and then put that plasmid back into the bacterium. And then from there, we can either make more stuff from the bacterium, like insulin, directly from the bacterium, or use the bacterium to transfer that DNA to other organisms, like plants, for making better food crops. So the point of the plasmid in this entire process is that it is the carrier of the new gene that we want to put into the genetically modified organism. So let's look at our answer choices here. Choice A, they are used to create clones. No, to create a clone, you would need to modify the entire genome of the organism, or I'm sorry, replicate the entire genome of the organism. So for the bacterium, it would be the circular DNA. For a plant, it would be the entire genome that's in the chromosomes. So no, plasmids are not used to clone organisms. Choice B, they are used as vectors to insert gene into bacteria. Yes, they are used as vectors, and vectors is just any method of carrying something. So for example, mosquitoes are vectors of disease. Okay. Choice C, they are a functional part of binary fission. No, they're not. They could be, but they don't have to be. So that's kind of iffy. So choice C is mostly wrong, I would say, but not as correct as choice B. Choice B is like, yes, that is the one. Choice D is they contain the circular chromosome of prokaryotic organisms. No, the circular chromosome is the genome, which is this guy up here that we talked about. So that's not the plasmid. So choice B is the most correct choice